Figure 3 consists of the results of cytoskeleton markers and transcription factors that demonstrate how the stiffness of the hydrogel affects the lineage specification. Figure 3a shows the immunostaining of the cytoskeleton markers and transcription factors with a hydrogel varying in stiffness from 0.1 to 1 kilopascal, 8 to 17 kilopascal, and 25 to 40 kilopascals. The imaging for the neurogenic matrices showed that for the softest hydrogel had the most expressed beta-tubulin. The imaging showed that for the minergenic matrices had localized the transcription factors for MYOD1, which represented by the white arrow, and for the osteogenic matrices upregulation for the transcription factors CBF alpha 1, which is represented by the white open arrow. In each matrices demonstrated that the stiffness of the hydrogel and the upregulation of the transcription factor are related. And then again in figure B3b, this is a microarray of the mesenchymal stem cells that were cultured in two different hydrogels. One gel was made at 11 kilopascals and the other one was at 34 kilopascals, and after the 1 week, the C2, C12 mydioblasts did not express significant MYDO1 on the stiffer hydrogels. Figure 3c is the fluorescent intensity of differential markers over varying stiffness of the substrate. And as we can see, this supports the findings from the previous two figures within figure 3, as it shows the average peak is within range of the stiffness to the immunostaining images, where neurogenic differentiation is related to a softer hydrogel between 0.1 and 1 kilopascal midiogenic, and C2, C12 midioblast differentiation is within the range of 8 to 17 kilopascals, and osteogenic and the human osteoblast differentiation is on the stiffest hydrogels. In figure 4, the plasticity of the mesenchymal stem cells are tested after 1 week if they can differentiate into another cell type based on the stiffness of the hydrogel they are placed into. So figure 4a and b show the progression of the differentiation on the appropriate substrate stiffness and the expression of the transcription factors. And figure 4c is a Western blot imaging of the expression of the transcription factors as it does relate to the stiffness. And then figure 4d shows that after the cell type has differentiated, after 1 week, that the cells will not differentiate into another cell type despite the changes within this microenvironment of the hydrogel stiffness. And that once this 1 week has passed, these cells are committed to their differentiation. And it also shows that the stiffness or the rigidity and signaling are needed to achieve the correct cell type. And so in conclusion, that the lineages of these stem cells and the differentiation is related to the microenvironment and stiffness of it, as well as the transcription factors, and that the stem cells are plastic until a certain point of progression within their differentiation, and that once that certain point has been reached, that they will not differentiate into another type, and so that they are committed until a certain point. And so for the experiment, we wanted to see whether if we can do an in vivo application of this. And so we wanted to focus on cardiovascular tissue and how the scarring around it is affected for the differentiation of these stem cells. And so the process that we were going to go through would be to culture these cells after extracting it from a mice, and then culturing it, that way we can get it to that 1 week marker, and then place it onto the damaged tissue to see whether if it would take. And what we would hope to see is that expression of the proper transcription factors. And the way we can test that is by doing a Western blot as well as through immunofluorescence. Another way that we can do it is by using RNA transcriptome sequencing, because we want to be able to see these transcription factors being openly expressed and that these stem cells have taken and are still continue to differentiate into healthy heart tissue. And so that way the scar tissue can be replaced by this healthier tissue. We would want to compare this method to just taking fresh stem cells and then placing it onto the damaged tissue, as the scar tissue that's going to be forming is going to have a different firmness to it, and this might inhibit the progression of the differentiation, as it's not going to have the same type of stiffness as like healthier tissue is going to be. And so what we're probably more likely going to see is that the cells that have not had that 1 week of differentiation on a hydrogel and then being applied, in comparison to the stem cells that are just cultured and then placed onto the scar tissue, is that the scar tissue stem cells are not going to properly differentiate due to this difference in stiffness of the tissue. However, we will see the differentiation continue in the hydrogel-based stem cells that have had that 1 week to properly get to that reaching point where they're no longer plastic and that they'll continue to differentiate into the proper cell type.